Hello friends, now we are going to continue with the cell cycle and cell division. Now it is about uncontrolled mitosis. If mitosis is not controlled, unlimited cell division will occur causing a cancerous tumor. So oncogenes are the special proteins that increase the chance that a normal cell develop into a tumor cell. Nothing but cancer. Cancer is nothing but uncontrolled mitosis technically. Coming to the next topic, meiosis. Meiosis is nothing but formation of gametes, eggs and sperms. Meiosis means reduction. In the meiosis, you can typically see, so there will be half the number of the chromosomes to the daughter cells. For example, if the parent cell is 46 chromosomes, the resultant, the cell division will have a 4 daughter cells. So each daughter cells will have 23 set of the chromosomes, which is half of the number of the chromosomes of a parent cell. Because of that, we call it as meiosis, means reduction. The facts about the meiosis is, it is being preceded by interphase, which includes the chromosomal replication. The event will be happening of both mitosis and meiosis. So there is nothing any change. The two mitotic divisions will be there. Two meiotic divisions will be there. Meiosis 1 and meiosis 2, which we call it as reduction division. Original cell is diploid, that is 2n. So we will have a 4 daughter cells produced and that are monoploid, means single n. Daughter cells contain half the number of the chromosomes as the original cell, produces the gametes. For the production of gametes, we need meiosis. Egg and sperms occurs in the testes in the males through spermatogenesis and occurs in the ovaries in the females through oogenesis. So more meiosis facts are, starts with 46 double-stranded chromosomes. After first division, 23 double-stranded chromosomes will be there. After the second chromosome, 23 single-stranded chromosomes will be there. Occurs in our germ cells that produces the gametes, nothing but in testes and ovaries. Why do we need a meiosis? It is the fundamental basis of sexual reproduction. Two haploid gametes are brought together through fertilization to form a diploid zygote. So fertilization, putting it all together, we have a haploid sperm and haploid egg. Two together are meeting to form 1n and 1n, we will have a 2n. So for example, 3 and 3 will keep up the 6. So this has to be continued to maintain the generations with the same chromosomal number in sexual reproduction. Replication of the chromosomes. The replication is the process of duplicating of a chromosome occur prior to the division, means during interphase. So the replicated copies we call it as sister chromatids held together by centromere. So the relation between this and this is, these are the sister chromatids held together by centromere, and these are the replicated chromosomes, and this is unreplicated chromosomes in the normal resting cell. Now it is been replicated into 2. So a replicated chromosomes is nothing but we have the homologs, means same genes with on different alleles. For example, it is the gene X. So these are the sister chromatids with the same alleles and same genes. These are, and these we call it as non-sister chromatids. So in the non-sister chromatids also we have the same genes but different alleles. Next, meiosis will form a haploid gametes. Meiosis must reduce the chromosomal number by half. So the fertilization then restore the 2n number. For example, from mom and from dad. So that will leads to child. For example, if it is 2n and 2n, this will leads to 4n. This is not, the chromosomal number is not maintained in the child. So it leads to too much of the chromosomes. And further to the next generation, it leads to too much. So meiosis reduces the genetic content. So this is the original meiosis will be happened. So this is the haploid and haploid combined together to form a diploid. So this is the right number. So meiosis. So this is the diploid parental cell, homologous, that will separate into sister chromatids will be get separated in the meiosis 1. So initially this is the process of interphase. Next we have the meiosis 1 and meiosis 2. Initially in the meiosis 1 itself, the cell will undergo reduction in the chromosomal number. For example, initially it starts with 46. In the first meiosis 23 and 23. Next in the second meiosis will be very similar to the mitosis. Nothing but same set of the chromosomes will be there. So these all are haploid. These are also haploid cells. This is haploid and this is haploid, and these are also haploids. And next, meiosis 1, which is a reduction division. Coming to the nucleus, early prophase, the chromosomal number will get doubled. And in the late prophase, the nucleolar membrane and nucleolus will disappear, and this centrioles will move to the opposite poles, and now the spindle fiber formation will get appear. So now this centrioles spindle fibers will get attached to the kinetochore. This is the metaphase. So if there is any middle alignment, we call it as metaphase. Now the splitting of the chromosomes will take place, and both homologous chromosomes only will be splitted. Splitting of the chromosomes, not the splitting of the centromeres. That the thing has to be notified in meiosis 1. For example, we have for example, assume we have 1, 2, 3, 4, 4 chromosomes. These 4 chromosomes are arranged in two alignment, two line alignment. Now it is been splitted. Now there is no any splitting of the centromeres has been taking place. The homologous chromosomes has been splitted into two daughter cells. That is it. So now this 4 has been divided into 2 to 1 and 2 to 1 cell. So the nuclear envelope is again reappeared and nucleolus is formed. So now the 4 has been divided into 2 to 1, one daughter cell and 2 to another daughter cell. This will be happened in meiosis 1. Next, what happens in the prophase 1? In the prophase 1 we have the meiosis. In the meiosis 1 we have the prophase 1. So it is again splitted into early prophase and late prophase. In the early prophase, the homologous pairs will be formed and crossing over will occur. In the late prophase, the chromosomes will condense and the spindle formation will occur, and the nuclear envelope will be formed in the form of fragments. Next, tetrads form in the prophase 1. So we already seen that in the prophase 1, so the homologous chromosomes will pair up. For example, this is the chromosome for hair color and body color from the father, and this will be the other chromosome for hair color and body color from the mother of a parental cell. So this pairing of the homologous chromosomes will be takes place in prophase 1. So this after pairing with the sister chromatids, each with, so these are sister chromatids, and the relation between this and this is non-sister chromatids joined to form a tetrad. This we call it as tetrad, and this whole process mixing we call it as synapsis. Synapsis means connection. So tetrad means having 4 chromatids. So tetrad means 4, having 4 chromatids. Because of that they named it as tetrads. And we also call it as bivalent. Bivalent is based on the terminology of chromosomes. Here we have 2 chromosomes, that is equal to bivalent, and 4 chromatids, that is equal to tetrad. Crossing over. What is crossing over means? The homologous chromosomes in the tetrad will cross one over each. So the pieces of the chromosomes or genes has been exchanged, produces the genetic recombination in the offspring. So this fragment will go to this chromosome and this fragment will go to this chromosome. So this whole process is called it as crossing over. Next, so homologous chromosomes during the crossing over, this is the picture. So this is the one more picture about the crossing over. So here the color has been changed. So crossing over multiplies the huge number of different gamete types produced by independent assortment. So it follows the law of independent assortment. Coming to the next, metaphase 1. The homologous will pairs of the chromosomes align along the equatorial line. So now after crossing over, the homologous chromosomes will pair up and align in the equatorial line. Next, anaphase 1. A for away. Homologous chromosomes will separate and moving to the opposite poles. So the sister chromatids will remain attached to the centromeres. Next, telophase 1. The nuclear envelope will reassemble, spindles will disappear, and the cytokinesis divides the cell into 2. So the same thing, whatever is happened in the mitosis, the same thing will be there. Next, meiosis 2. So only one homologous of each chromosome is present in the cell. Okay, only one homologous chromosome. So now the sister chromatids carry the identical genetic information. So the meiosis 2 produces the gametes with one copy of each chromosome and thus one copy of each gene. Now in the meiosis 2 is nothing but the splitting of these homologous chromosomes will happen. Nothing but splitting at the centromere. This splitting of the centromere will not be happened in meiosis 1. Next, meiosis 2, reducing the number of chromosomes. So this is prophase 2. So meiosis 2 is again divided into prophase 2, metaphase 2, anaphase 2, telophase 2, and forming of four identical daughter cells. So initially we have 23 and 23. Initially it is started with an interphase of 46, and we have the meiosis 1. In the meiosis 1 there is no any splitting of the centromere and no any splitting. So the 46 has been divided into 23 and 23. Now we have 23 and 23. Later, these chromosomes, whatever the same thing will be in the mitosis. So this 23 and 23. Now the splitting of this homologous chromosomes will take place. So now each cell will go for an identical number of chromosomes. So again all the four will have 23, 23, 23 and 23. Here the splitting of the sister chromatids will take place in meiosis 2. Coming to the prophase 2, the nuclear envelope will fragments and spindle forms. Metaphase 2, chromosomes will align along the equatorial line. Anaphase 2, so the pulling away, A for away, so the sister chromatids will get separated and move to the opposite poles. And next telophase 2, the nuclear envelope will reassemble, the chromosomes will decondense, the spindle will disappear, and the cytokinesis divides the cell into two. Next, result of the meiosis. Gametes, eggs and sperms are get formed. We have the four haploid cells with one copy of each chromosome, one allele of each gene. So the different combinations of the alleles from different genes along the chromosomes will be takes place. So gametogenesis, oogenesis or spermatogenesis. Coming to the spermatogenesis occurs in the testis. The two divisions will produce four spermatids. So the spermatids will mature to form sperms. Man will produce about 250,000 sperms per day. So coming to the spermatogenesis in the testis, so first it is spermatogonium with 46 chromosomes, and next, so which will be on differentiation, it will undergo meiosis. So with the 46 chromosomes, instantly it will start, and it will, after the end of the meiosis 1, 23 and 23 is being splitted to the sister chromatids. Now and again it will divide into four cells. So the same meiosis 2 will be happen. This 23 is being divided into two identical cells, and here we have two identical cells. Here the homologous chromosomes will separate. Here the chromatids will be separated in the meiosis 2. So spermatogenesis. So in the spermatogenesis the same thing. So autosomes, allosomes. So one side in spermatogenesis 50% of the sperms will have the X chromosome and 50% of the sperms will have Y chromosome. Next, oogenesis. It occurs in the ovaries. Two divisions will produce three polar bodies and that die and one egg cell. So the polar bodies die because of unequal division of cytoplasm. To make it clear about this oogenesis, go for the video of the menstrual cycle and fertilization. Then the oogenesis will have a clarity about the cell division. Oogenesis and cell division, in what happens in the oogenesis, and what are the three polar bodies, what is only one egg cell. In oogenesis, the very characteristic feature is one cell will give only one egg cell. That is the very important thing about oogenesis. But one spermatogonia will give four sperms, but one oogonia will give one egg. So the polar bodies will die because of an unequal division of cytoplasm. Immature egg cell we call it as oocyte. So starting at puberty, one oocyte matures into an ovum for every 28 days. So oogenesis in the ovary, this is the thing. Initially it is oogonium. Oogonium will divide into primary oocyte. Now the primary oocyte will divide into first polar body and secondary oocyte. Now the secondary oocyte will undergo into egg and polar body. And these polar body will again divide into two polar bodies. All these three polar bodies will die because of unequal division of cytoplasm. And only this one egg cell will fuse with the sperm to form an embryo, zygote. So only one egg cell is being formed from one oogonium. So this is again about the oogenesis, chromosomal number. So comparison between mitosis and meiosis. Coming to the comparison, the number of divisions happen in the mitosis is 1, but in the meiosis, 2 divisions. The number of daughter cells is 2 in mitosis and 4 in meiosis. Genetically identical, yes. Here there is no any genetical identity. Because of that, we all the sisters or we all the brothers, siblings will not look similar because of this meiosis only. because of the crossing over which is happened in the prophase 1 of the meiosis 1. Chromosomes, same as parent. Chromosomes, half of the parent. It happens in the somatic cells, it happens in the germ cells. It happens throughout the life. It happens during the sexual maturity, role, growth and repair. Here it is sexual reproduction. Yes. So this is all about the mitosis meiosis and its differentiation. So thank you very much. Please do like, share, comment and subscribe to my channel.